Let's try to understand now the cell cycle. The cell cycle is a term refers to the events happening during the life of the cell. And we can divide these events into two periods. The first period is called the interphase, and the second period is the cell division or the mitotic phase. The interphase constitutes most of the cell life. All of these are the interphase, whereas this is the mitotic phase. So as you can see, most of the cell life is happening during the interphase. This is where the cell is performing normal function. So for the interphase, we can divide it to three subphases. The first one is something called the G1. G is coming from gap, so this is gap 1, and during which the cell is growing. So all of the normal growth processes are happening during this G1 phase. Most of our cells are in G1 phase, some of them in G0. But anyway, either G1 or G0, it doesn't matter. Most of the cells are in this phase. So this is when the cell is performing its normal functions. If it's muscle, it will contract. If it's neurons, will send signals. If it's a cardiac muscle, it will contract, and so on. Sometimes the cell will decide that it needs to divide, and when it needs to divide, it will move to the second phase. The second phase is the S phase, and S is coming from the synthesis. During this phase, the cell will replicate its DNA. So DNA replication is happening during this phase, and we already talked about this. S phase is only for DNA replication. After the S phase, the cell would move to gap 2. And in gap 2, the cell will perform its final preparation for division. So it can make more ATPs, it will make the enzymes it needs during division, it will make the organelles that it needs during the division. Whatever the cell needs during division, it will prepare during the G2 phase. After that, the cell will move to the M phase. Now, before going into the M phase, we need to understand that M phase is about splitting the DNA. Remember earlier when we said we are going to split the DNA, we are going to replicate it and split it into the daughter cells. This is happening during the mitotic phase. And this is, we will see it's a very critical process, because any mistake during this phase, it means failure. And the daughter cells might die or might carry a very significant mistake that will cause them to be cancer cells or problematic cells or initiate some diseases. So the mitotic phase is a very critical process. And the cell would try to keep all of its resources for the M phase. So during the M phase, the cell is not ready to make any ATP, it's not ready to make any enzymes, anything. All of its focus on the M phase. That's why during the G2, it will make the final preparation for the cell division, meaning that it will do all of the ATPs, do all of the enzymes, do all of the proteins that it needs. So let's see now the mitotic phase. The mitotic phase is basically made out of 4 stages. These are prophase, the metaphase, anaphase, and telophase. BMAT. Prophase, metaphase, anaphase, telophase. And they are always in this order. BMAT. During a prophase, the cell is going to lose the nuclear envelope. Some of the organelles will start to disappear. We can see the chromosomes within the center of the cell, and we will have 2 centromeres. These centromeres will start to go away from each other. All of this will happen during the prophase. Then the metaphase. In the metaphase, we will have the chromosomes lined up in the middle of the cell. The centromeres are far away from each other, and they are already formed what we call it the mitotic spindle. The mitotic spindle is a device that will help splitting the chromosomes. This mitotic spindle will bind to the chromosomes. All of this will happen during the metaphase. Then we will have the anaphase. In the anaphase, the chromosomes, or now what we can call them chromatids, they are separated from each other. And each set of chromatids is being pulled toward one end of the cell. It's being pulled because of these mitotic spindles are shrinking now. So they are bringing these chromosomes toward one pole or one end of the cell. This is the anaphase. Then there is the telophase. In the telophase, the chromosomes are already at one end. they are far from each other, they will start to deassemble. So they are going to form the chromatin again. The mitotic spindle is shrinking. We will start to see some of the organelles in the cell. The cell itself is elongated, and it's ready now to do the division of the cytoplasm. And the division of the cytoplasm is something called cytokinesis. Cytokinesis, it's the final cut between the two cells. The cytokinesis is the one that would establish two independent cells from each other. So they will be cut, and each one now is a separate cell, that it has its own nucleus with its own nuclear envelope. The chromatin are inside. It has the full organelles, the full sets of organelles as its mother cell. This is the cytokinesis. And usually cytokinesis would overlap with telophase. So this is an overall summary of these steps. You need to know them. Maybe you don't need to worry about the pro-metaphase. Just know the prophase, metaphase, anaphase, telophase, and of course the cytokinesis, and a brief description of what happened in each one of these steps. This concludes chapter 3. Please post your questions into the forum. If nobody could answer them, let me know and we can go over them together.